All right, very good, everyone. Welcome. We will go through introductions first, and let's start with, at the far end, with my colleague, Leisha Sequist.
Hi, everybody. Is it already on? My name is Leisha Sequist. I am a medical oncologist. I focus on lung cancer and early detection. I work at Mass General Brigham Cancer, and it's really nice to see you all.
Good afternoon, all. My name is Gianluca Pettiti. I'm with Thermo Fisher Scientific. I run our technology businesses. For the one unfamiliar with the company, we have 120,000 colleagues. We're a world leader in serving science, both in the pharma, biotech, and diagnostic space. I look forward to spend time with all of you today.
Good afternoon. My name is Eben Chen. I'm also a medical oncologist at Mass General Brigham, and I have the privilege of directing the bone marrow transplant and cellular therapy program there.
And hi, everyone. I'm Dave Ryan. I'm the president of the Mass General Brigham Cancer Institute, charged with bringing everybody together across the Mass General Brigham system around cancer. And by training, I'm an experimental therapeutics and GI person, mostly colon and pancreas. And over the years, I've taken care of a lot of folks with colon and pancreatic cancer and still do. So today's session is really about setting up the academic medical system to treat, prevent, and cure cancer of the future. We're in the process of doing it, and what I wanted to do today is center the conversation around two stories. The second story is going to be a cell therapy story, but the first half, I want to center on a story that's been a dream of mine for many, many years as a pancreatic cancer doc, and that is a 50-year-old man comes in to his primary care clinician, who is 10 years from now, is a nurse practitioner, and the nurse practitioner is seeing that particular patient, and up on the screen pops an algorithm generated by one of our programs in machine learning, either at our place or another place, that says, hey, this patient has a 50-fold increased risk of having pancreatic cancer in the next 5 years. At that visit then, a multi-cancer early detection panel, and we can talk about what that panel looks like, is drawn, and sure enough, the cart's put before the horse, and there is a pancreatic cancer DNA, proteomics, epigenetic profile that says this person has a pancreatic cancer. There's also a research study that has recently identified a radionuclide PET scanning imaging agent for pancreatic cancer. Identifies a 3-millimeter focus in the tail of the pancreas. And then there's a consult with the radiation oncologist and the surgeon, and a decision is either made to ablate it or to do a robotic pancreatic tail resection, which you're out of the hospital now in 2 days. And the punchline is that that person never meets me and never dies from pancreatic cancer at the age of 55 like they were meant to. So all of those technologies exist today, we think, in nascent forms. We've seen the future. So let me turn to Leisha and tell us what aspects of that story come to mind first around setting up the Cancer Institute of the future.
Yeah, this is a really hopeful story, but one that I don't think is far-fetched. I think this really is just around the corner for us. A couple of the key insights about this story in particular. One is that we're monitoring someone's health, or in this hypothetical medical system, they're monitoring someone's health using everyday data in order to try and understand better what risks that person faces. And that's really something, we need to do more in the cancer field, is to understand out of the whole population which people should we narrow our focus on for which cancers. Because they're not always what's on the top of the mind for the patient. The patient may be thinking about, I'm at risk for breast cancer because my mom had breast cancer, but that it turns out that maybe their blood sugar levels are more indicating pancreatic cancer. And it's really hard to keep all of these different algorithms in mind as a human provider, primary care practitioner. So having algorithms that can help lead us towards risk and really focusing our screening in a precision and prevention way, I think is really important for the cancer centers of the future. And then secondly, if an algorithm or some kind of test flags someone as being high risk, we have to have specialized tests that we can do. Tests that maybe don't make sense for the whole population to get all the time, but once you're in a high-risk group, how can we develop, with a variety of different technologies, the best kinds of early diagnostic interventions to find those tiny, 3 millimeter, 5 millimeter cancers that can be highly curable.
So one of the follow-up questions to that is, in terms of the test, and maybe I'll turn to Gianluca about this, there's 3 broad applications, I would say, right now. One is looking at genetics themselves circulating in the bloodstream. The second is looking at epigenetic processes like fragment size or methylation. And the third is proteomics, which has gone recently from analog to digital. Do you think one is going to emerge, 2, or all 3?
Well, as the only engineer on the panel, I think I'm surrounded by medical doctors. I use a technology example, and is the self-driving car. And I think collectively we're building a self-driving healthcare or medicine, or that's at least the hope. And very much like in building a self-driving car, you can't rely on one variable. You actually need a combination of different variables, and obviously biology is a lot more complex than having a car driving itself. And so I think the answer is all of the above and some more. I had the opportunity to experience face age and AI-driven algorithm at the MGH that is inferring the biological age from the image of your face. I won't disclose what the result was for me, it's confidential. Enough to say that this AI algorithm, they need some tuning still. I'll leave it there. But the point being, when you look at things like the UK Biobank and the findings that we're seeing, specifically in proteomics, with now publications that are showing how some of these multi-protein signatures are present in individuals as far as a decade before a cancer diagnostics is released. It's quite impressive. And again, compared to the analogy of a self-driving car, it means that instead of letting the car crash and then fix the car, you're actually preventing that. To me, proteomics is like the GPS, genomic is the map. Together, they have the potential with epigenetics, and then undoubtedly with the advancement of healthcare and the adoption of AI to truly transform the way that we drive not only the understanding of disease, but early diagnostic and then early intervention. I think a big question for all of us is what's going to be the education required for not only the medical systems and technologists, but also for patients to be able to take full advantage of those technologies, because I still have my wife that has a Tesla that is self-driving, and she said, no, I don't trust the Tesla, I'm going to drive myself. And at times I say, maybe you should trust the technology a little bit more.
I mean, in your analogy to the taking care of the car before the car crashes, I think is timely. I mean, in the time I've been in practice, our entire system of seeing patients and being physicians has evolved greatly, and that includes how we're all taught to be physicians. I can guarantee we were all taught to be physicians by asking a patient, what's your chief complaint? Meaning they show up with a complaint. And that's all geared around taking care of the sick. But what we're talking about here is actually taking care of the well, understanding your risk going forward, and then how to modify that risk. And that's a complete change in the paradigm of how we practice medicine. And you're right, not only do we have to learn the technology, but how we educate the future of healthcare has to be there as well.
So this has multiple issues that get raised, and how do you manage patients within an academic medical system around wellness? And you have folks who are scared that they might have cancer. You have folks where the primary care physician is scared that they have cancer, that maybe that there's a mass. And then you have probably the best predictor of future cancer, which is germline genetics. And so Leisha runs our early detection and diagnosis clinic, and we've made some decisions recently. One, housing it adjacent to germline genetics, but why don't, Leisha, you talk about some of the issues that are getting raised around the primary care community and multi-cancer early detection assays, and how you're thinking about designing this clinic of the future.
Yeah, it's been a really interesting learning curve. We launched our clinic about 2 years ago, and we've been growing steadily because I would say that both patients and primary care doctors are really interested and yearning for more collaboration, not waiting until a patient has a diagnosis of cancer to reach out to an oncologist, but thinking about pre-diagnostic steps and can you figure out who's at risk. So, we offer consultations about cancer risk assessment, in which a very key part of this is germline genetic testing. We've been really surprised at the high level of germline mutations that we've seen in testing what you would think of as a healthy, population. The literature out there suggests about 1% of us walk around carrying one of these germline mutations, but we've actually found consistently now over a couple of years, 11% of our patients are positive. And so I think it just that this testing was so limited in the past, we didn't fully understand the scope of the problem. The more we start testing the population, I think the more we're going to realize how much of these risk factors may be out there. But germline genetic testing is not the only type of risk factor. The other thing we've really learned in our clinic is that, these new blood tests that are coming out and are coming to market, some of them are available pay out of pocket. They're not yet covered by insurance. But these tests that promise to be able to screen for multiple types of cancer at once are very appealing in some ways to patients, but very daunting to the way that primary care is set up in this country. The primary care offices are just, for the most part, not set up to counsel about genetic tests, cancer genetic tests. They're not set up to interact with the different vendors that are providing these different tests. And the hardest part, I think, is interpreting the results. The results come back with a lot of gene names, and we found that the primary care doctors are really, really excited to partner with us because as oncologists, we have a little bit more familiarity with thinking about cancer genetics. It's part of our therapeutics. It's part of the testing that we do routinely on patients diagnosed with cancer. So it's been a really great partnership, and I think has inspired us to expand our program even more, to bring cancer specialists right into the primary care circle before people have cancer.
So if people have questions, please put them online and we'll get to them. I want to get to the second major change that's happening in our academic medical system of cancer care, and that's around cellular therapy. So cellular therapy has grown dramatically. And let me tell you about a current story. Instead of a future story, let me tell you a current story and the implications that it has for us in terms of setting it up. Eben runs our cellular therapy service clinically, oversees what we call the cell therapy pharmacy, which every system needs a place for cell therapies to get collected, the cells to get collected and go out to whatever the lab is, whether it's an academic GMP facility or a company. And another place to receive those cells coming back in. And most places, including ours, this developed out of the bone marrow transplant program. So there's an ongoing clinical trial at Mass General Brigham, which you can read about, the ASCO update. It was in the New England Journal, the first 3 patients, and the ASCO update was this past June, of an ongoing story in GBM. But let me give you a little insight into how that all started. So Marcella Maus and Brian Choi created a CAR T that targeted EGF and then coded it to secrete a bispecific at the site of the tumor. And they came to me and said, hey, Dave, we want to inject this into the brain. I'm like, you can't do that. And they're like, no, we can do it. Everything will be okay. And I'm like, you're going to kill somebody. And they said, no, we're not. Brian said, I'll sleep overnight. He's a neurosurgeon. If they need a shunt, I'll get the shunt in. I'll alert the OR. We'll alert the PACU. We'll alert the neuro ICU. Eben's team said, Dave, no, we got this, we'll sleep overnight, and which they did. Mark Like slept overnight. The incredible nurses who are used to dealing with cytokine storm up there in our bone marrow transplant unit were right there. Eben's team was amazing. The neuro-oncologist said, you know, we got this, we'll work together with the cell therapy team. And sure enough now, we've treated a number of patients, over 10, and it's been an amazing effort. But the two things that came out of this, in my book, which changed everything from here on out, which is that, number one, you can inject these into the CSF. And so people like Merit Sukovich, who runs our Neurosciences Institute, is an ALS expert. The MS folks were interested. All the autoimmune folks were interested in the CNS space. The second is that it's no longer about targeting, like in heme malignancies, where you target a CAR and they've had this, they target a specific antigen and have this incredible anti-cancer effect. But in solid tumors, it's about delivering what you want to the local tumor microenvironment. And think about all the different things that you can deliver. And as a place of research and innovation, yes, you could argue that down the road, bispecifics might be just as effective, but are they going to be? And is that the right place to do the initial clinical trials? So I'll stop with that part of the story there. And let's hear from Eben about how this has affected the way you think about setting up cell therapy at our institution.
Yeah, I mean, it's an amazing story. I'll just say, before I answer your question, I'll just reflect and I'll say I'm happy for a couple of things. So I'm happy that I've been in practice to watch the evolution of this field and how amazing it was. We knew that white cells killing cancer was an effect that was here to stay. It's just a question of how we're actually going to manipulate, cultivate, and do it. I'm happy that I could hire young and talented, we could hire, he has the money, that we could hire young and talented people to actually do this work, and we got them here to MGB. I'm happy that Mark Like, who stayed overnight, wasn't married at the time, so he could stay overnight and watch that patient for like several nights in a row. But we were worried. I mean, it was in the early days of CAR T, we didn't treat patients with tumors in the brain. These are blood cancers because we were worried of the brain toxicity that would happen. And this was actually injecting those CAR T cells directly into the cerebrospinal fluid, right? Because your body naturally has a blood-brain barrier that blocks a lot of white blood cells from getting there. So that was the big obstacle. But if we could inject those CAR T cells directly into that space, number one, then you might actually see some therapeutic activity. And the second thing is what Dave said is with the evolution in gene editing capabilities, we're then able to take these cells and not only express a receptor where you can target them so they know where to go, but in such a cool way, you can now make these cells do other things, right? So you can make the cells target there, and then the cells can secrete all kinds of things that they need to make their activity more effective, to make the surrounding environment less immunosuppressive, to make them last longer, to recruit their friends, I guess, other white blood cells to the environment to carry on that activity. So it has revolutionized how we think about cellular therapy, because not only, right now, most of the commercial standard of care cell therapy, as many of you know, is all approved for blood cancers. Okay, that's the majority of the market. But with this success and other stories, we're able to think now about moving into not just solid tumors, but we have this whole lineup of non-oncology studies that we are approving left and right to start treating non-oncology conditions as well, to use cells as a living drug to modify those effects. And the ability to be able to not just deliver it safely, but then gene edit to make it do what you want it to do, that's going to be super powerful going forward. A lot of challenges, which we'll talk about, but it really has opened up that entire box of how we're going to do this.
I have two follow-up questions. The first is, what are the rate-limiting steps in the system for you to be able to do this? And the second is, how is industry starting to respond? And maybe I'll ask Gianluca to step in on this one too. How is industry responding to the cell therapy issues that are arising, whether they be this potential for in vivo or this pushback on, well, bispecifics can do the same thing?
So I'll hit two rate-limiting steps first and hand it over. One is that most of the cell therapies we use now are individual, meaning they're one-off drugs made for each patient. And you can understand, one, how much time that takes from a manufacturing, or how much effort and cost that takes from a manufacturing point of view, and then the subsequent expense that is to the patient, the healthcare system in general. So that approach is probably not sustainable from a scale-up model. Two is the regulatory pathway to get something approved, right? So many of you may have heard about the story at UPenn where they diagnosed a newborn baby with an inherited defect in the urea cycle. So they built up proteins, and without a liver transplant, that baby would die. Even with one, they would still have issues. And from 1 month to 7 months, they had developed a vector and a gene therapy that they could give the baby to correct that mutation. A gargantuan effort that was published and really awesome to see. The issue is that, these inherited diseases are rare, and to get one of those diseases approved, the amount of investment in R&D necessary in the regulatory pathway is huge. You're all aware of this, right? If we're able to actually not look at the specific mutation itself, but perhaps the platform or the device or the vector or the delivery model used, and then can exchange those things, then we'd be a lot more efficient. But there's a lot of challenges there, and so those are my two big challenges going forward.
Yeah, maybe reacting, obviously aligning with the perspective 100%. These are areas we're spending a tremendous amount of time on. I wish we were further along the journey, but I'm encouraged by the fact that we're seeing more standardization in the approaches with the great work that you're doing and many of colleagues of yours are doing as well. We start to see more standardization, and our role is try to embed that standardization into technology platforms that then can be adopted. Our approach at Thermo Fisher, as an example, has always been to try to have a modular workflow to actually allow in the first phase of cell therapy to be able to customize workflow that really works. Because trying to standardize something that is not 100% understood, it's not a good idea. But now I think we are at the point where we're seeing more standardization being possible, which will over time drive, hopefully, the cost to a level that is more sustainable. We're seeing some of these therapies in some of the emerging market right now because of necessity to become quite affordable. India was in the news recently with, I think, $20,000 cell therapy. And so obviously there's opportunities. I think the regulatory aspect is a very complex one that will also get the benefit of more platformization of cell therapy. And then hopefully over time, we're going to see less need of the N of one and a better understanding on how we engineer the immune system in order to have truly allogeneic solutions as opposed as autologous, which I think ultimately will need to play a role over time. But there's a lot of work to be done, a lot of technology to be developed, and we're fully committed to do it. I wish we could go a little bit faster.
Let me ask about the in vivo CAR. Can you describe what that is? And this came up at our institution around, do we invest more heavily in a GMP facility or not? Most people don't understand the difference between a cell therapy pharmacy and a GMP facility, so maybe start there. And then the second is, well, we're not going to need to invest because these are all going to be in vivo.
Okay. So a cell therapy pharmacy or a cell processing lab is what many institutions who were doing conventional bone marrow transplant had. This is a place where our technicians and our scientists can handle cells. They can process them, they can freeze them, they can thaw them, and get them ready for infusion into patients. A GMP facility, GMP stands for Good Manufacturing Practice, is a facility that meets all different, much more stringent regulatory requirements that allow you to manipulate cells beyond what the cell therapy pharmacy can do. So you can do the If you have your own GMP facility at an academic institution, then you can actually manufacture your own constructs with gene editing and so forth to make your own CARs or other cell therapies. Whereas a cell therapy lab, you're just able to process them and prepare them for infusion, okay? Right now, the vast majority of cell therapies that we talk about, the CAR T cells you hear about, they're all in vitro or ex vivo. So what happens is I see a patient, we collect their white blood cells or T cells, we send them to a company, the company takes those T cells, makes them grow, activates them, and then uses a gene editing technology to insert whatever gene they want, usually a receptor, and then packs it up and sends it back to us, and we infuse that into the bloodstream of the patient after a little bit of chemotherapy. That's modern CAR T cell treatments. What Dave is talking about is in vivo. So what happens if you can bypass that whole step of sending it to the company, number one, and also waiting the 3 to 6 weeks or longer it takes to manufacturing that product, all right? What happens if you can just treat the patient by injecting them with a vector or a delivery system, and in that vector is the gene editing technology. So you can just inject it directly into the patient, and if it has the right vector that knows what cells to seek out, and it has the right gene editing machinery, it knows how to change the genes in the body itself, then you can skip that entire step. So that's the hypothetical promise of in vivo cell therapy, is that we can skip that whole thing of taking the cells out. A lot of challenges. There's some trials going on. In vivo is a lot more complicated because you're injecting it into the body, and the body has so many more cells, right? In vitro, you have the cells you want, you can purify them, and you have full control over what you're doing. In vivo, you send them into the patient, and you hope they find the right cells, that they're inserting into the right part of the DNA, and then your magic happens. The other part of in vivo is that the body has an immune system. It's not going to like that stuff. And then the last part is you inject in vivo. So the CAR Ts we're building now, they're blood cells, and then we inject them back, and they persist for a period of months to even years, okay? Because we know how to do that. If we do in vivo gene or cell therapy, or in vivo gene therapy to be exact, you insert the vector into the patient, you're not changing their DNA in all their cells. It's just a small portion of cells that that vector can find. How long is that going to last? Those cells don't live forever. So if you're trying to fix a genetic defect, right, are you going to then commit this patient to lifelong infusions repetitively because those cells will ultimately die, and your body will ultimately reject that vector that you're using? So these are the challenges that we talk about when we think about, can we skip that step and do this in vivo gene or cell therapy?
So before we get to online questions, and there's a couple, I just have a follow-up question for Eben. Beyond cancer, where do you see the greatest uptick of cell therapy?
I mean,
in broad-based diseases, obviously
Yeah.
the N of ones are going to continue to come up,
Right.
like the urea cycle story. But for broad-based diseases that the population suffers from, where outside of cancer?
I mean, right now it's all autoimmune disease right now. It really is. We are flooded with trials looking at how to modify autoimmune disease because the blood gives rise to the immune system and so forth. And part of those immune inflammatory diseases are the neurological diseases that Dave mentioned in terms of ALS or MS. And so, those obviously have a huge burden on the population. And so it'll be really exciting. I think we expect an FDA approval for an autoimmune disease in the next year or so, and that'll get us started. And then as we've made CAR T cell therapy safer and safer, it's interesting. In cancer, the stakes are so high. And so we say, let's do CAR T cells, and the lymphoma doctors are like, sure, let's go. They read the press, saw the toxicities, were like, yeah, let's go. And the patients are like, let's go, because this could be a cure. You talk to a rheumatologist who cares for the lupus patients or rheumatoid arthritis, and they have about 8 other treatments that can modify disease but not cure it, right? They can keep patients going, yet the burden of the the toxicities of the treatment as well as the morbidity of the disease continue, we talk to them about that. They're like, oh, wait, and they are really hesitant to put their patients to a treatment like this. And part of it, I get it. They have other treatments, and the stakes are a little bit lower than when you have a diagnosis of cancer. So it's going to be really interesting as we go forward in the next few years, as the trial results are published, to see how this changes.
From a systems perspective, do you trust your medicine colleagues to learn how to manage cytokine storm, to do preconditioning regimens, to give Flu-Cy? Or do you think that your cell therapy team writ large, including transplant, is just going to be called on more and more for an episode of care that get these folks through this 2 to 4 week window and then send them back?
Yeah, no, I've talked to my cell therapy team. We've accepted that we're not oncologists for a lot of our lives. We went into oncology, but we're going to be the modality of cell therapy specific type, and we're going to run a core type of facility or service that helps the other services conduct these trials. If there is a successful trial for, that becomes, as it moves earlier in lines of therapy, then I think that will be the only way that our colleagues will get interested to learn how to do this. But for right now, I think it's a fairly complicated, specialized niche of care that we're committed to collaborate entirely on to make it work.
All right, so let me get to the online questions. So the first question is about germline therapy. How do you select from those healthy populations who need to be tested? I'd say that, I'll turn this to Leisha, but the first thing that we're doing is creating universal germline testings for people with cancer. So for every solid tumor patient, and also some liquid tumors in there as well, but for the solid tumors, roughly 5 to 10% of all solid tumors carry a germline predisposition. Even your famous cancer, EGFR mutant lung cancer, which you're famous for, has about a 5% risk of germline predisposition. That's a 1 in 20 chance. And the people who are at most risk of one cancer are people who've already had a cancer. So creating germline availability for everybody with cancer is an enormous task. We see about 25 to 30,000 new patients with cancer at MGB every year. That means that we have to test all 30,000. I'm a doctor, so math is not my strong suit. So 30,000, right? So 10% is 3,000. Most of these, almost all of them, are autosomal dominant. So now you've inherited half their families for the rest of their life in a clinic that you are going to employ your early detection. And now we're talking about universal germline for risk. That is an enormous task. How are you thinking about it?
Yeah, I mean, I think that we have to prioritize people who maybe have family histories of cancer initially, but as we learn more, and as we learn more from our testing, universal testing of patients who've already been diagnosed with cancer, I think we may understand better the scope of the problem. A lot of the older data about, say, BRCA mutations in these families with just very, very penetrant genes and a very, very high risk of cancer, and Li-Fraumeni, that's not what we're talking about in 11% of the population, 20% of the cancer population, but it is maybe a more modified but still elevated risk of cancer. And I think that if we're going to find cancers in an earlier stage, we've got to start providing long-term preventive and surveillance care for those populations. So we have to build our system to accommodate.
Can I add one thing? So this is about is maybe more of a sociological or psychological point. That is, what is the true driver for an individual to go seek a diagnostic or a prognostic, like a data point that helps that individual to prevent something? And I fundamentally believe when you look at compliance with medication, there are life-saving medication, and you look at 50% compliance, you understand that there's something that gets in the way of individuals to test themselves. And so to me, payers will play a big role here, or they need to play a big role in the prevention part of the equation. And we're seeing countries in Europe where you have single-payer system, governments that are being active in actually creating large biobank, large population study to actually then help stratify risk across the population, and obviously with a fast-aging population in some of the European countries, including my own, Italy. I was in Greece recently. They really have an incentive in playing an active role. So I think the payers will need to figure out how they play an active role. It's like no one would insure our house without having a smoke alarm. So what is a smoke alarm for health? To me, is again, a technological question, but also a payer question and a health economic question that will need to be connected with the discussion that Lisha was highlighting in order to unlock it at scale.
We have time for one last question, and I'll finish with Eben. We answered most of the questions, but in addition to expense and time, it looks like it needs a lot of caregiver expertise, cell therapy. So how are you expanding these therapies to many hospital and larger community hospitals that don't have an academic medical center or system available to them?
That's a great question, because we talked about this at a level of once you start bringing in solid tumors or non-oncology conditions, which are much more common than blood cancers, and once there's a much more common approval, the number of patients needing or indicated for cell therapy will overwhelm every academic center's infrastructure, their lab, their capacity, and so forth. So it's not just it's not just when or if, it's when. It's not just if, it's when. We have to expand some of these therapies to the communities. And so how do we do it? It doesn't make sense for every community site to reinvent the wheel. It doesn't make sense for every community site to have a GMP facility. It doesn't make sense for every community site to have what we have right now to conduct cell therapy. So we have to figure out what we can build horizontally across the system or systems, all right, and bring that in. So if say our colleagues at, let me just use an example, Maine Medical Center, who we collaborate with a lot, say our colleagues at Maine Medical Center want to start doing CAR T-cell therapy, which is not far from a conversation I've had, you know, how do we do that? So we've gone up there and visited their site and said, this is what you need to conduct cell therapy. We can help provide this, this, and this to work with you, all right? I'm sure there will be at least some point some commercial interest that develop this type of horizontal structure to be able to do this, but we can provide this, this, and this. You already have the physicians. We can train your caregivers. You can come down here and do the caregiver expertise, but it's all of the other infrastructure. So where's your cell therapy pharmacy going to be? Where are you going to store products? How are you going to negotiate each and every contract that all these cell therapies have with each institution and the payers? And it's that entire sort of non-patient facing work that we have to find a solution for to make more efficiently. So that's the key, to be able to move that out to the community.
Well, we're at time, so I want to thank Lisha, Gianluca, and Eben for great answers to a wonderful panel, and I'm sure they'll be available to have conversations with in the hall. So thank you very much.
Thank you.